dosage that they give you.
But why would you want to take so much antibiotics?
Why do you want to do that?
Shouldn't you stop as soon as you feel better?
Because you're leaving behind the resistant ones to be selected for more resistant bacteria. So you're not killing all the bacteria that form the infection.
So what she's saying is that you might be feeling better, but there still could be bacteria inside of you that is resistant to it. So if you keep on taking the antibiotics until you kill them all off, it ensures that they won't become resistant to the antibiotics next time you get sick again.
Yeah, it's like you kill 99.5%, and that other small 0.5% are the resistant kind, and then they're allowed to propagate. Even though they're not making you sick at that point in time, you'll spread it to other people. They just get out to the environment.
Make sense? Yeah, because it's sort of calculated how long it takes, how much, what dose it takes to pretty much kill all of them. And if you stop, as they said, that small portion that was partially resistant maybe, that's kind of still, they're hanging there, but they're not doing so well, they're the ones that are going to then stay around. And now you're doing selection. Remember how we selected these butterflies? And so then the next generation of that population is a little more resistant. And then they can spread it to other.
But I think too, physicians, they're too quick to prescribe medications because the drug companies are pushing it. I mean, new drugs that really work well, good antibiotics, they're prescribing them for things that they really shouldn't be prescribing them for, because you don't need a WMD for some of these bacteria, but doctors are prescribing really strong bacteria, doesn't it?
You have
it
to
accelerates the process.
You have to be careful. This is the kind of the problem that the society that we're living in. We can't trust anybody anymore. That's the problem. It used to be, okay, I didn't used to trust my mechanic. I had to be careful because this guy may just tell me this is broken, but it's not. But today you can't trust your doctors either. It's like, who do I go to? This guy says, you take these, and then, and yeah, he's right. You have all kinds of pharmaceutical companies that are doing all kinds of things that shouldn't be done because there's money involved. All these commercials that they're doing now on TV, ask your doctor for this, and if you eat this, you're going to die, and if you eat that, you're going to, all kinds of other. But we have to be careful, though. It's difficult. Because if it's somebody who's expert, I normally want to trust that expert and listen to them. And if they say you need to take it, so I don't know any better, But he's right. There is some issues. The other thing is we don't test. We just, a lot of times, even the consumers, this used to be a bigger problem in the past than today. You would go to your doctor and they tell you, well, you have a cold, go home, you'll get better. But people wanted something. And a lot of times this is what? Viral infection. So if it's a viral infection, we didn't used to have anything for it. They will give you antibiotics. For whatever you needed, they will give you antibiotics. And that was another problem. Today they're training physicians to not do that as much as possible. Give them some sugar pills, something. Send them home so they're happy. And if they say this is viral, then you need not to ask. Okay, unless if they give you viral medication. Yeah.
Yeah, I agree with you too. I mean, I think that they should mandatorily test people if they want to get antibiotics because it actually helps. Because you can figure out what kind of bacteria it is within 24 hours. And I mean, starting antibiotics, if it's not a life-threatening condition, 24 hours later, it's not gonna kill you. And that way, know for sure.
In the best of situations, it's good to figure out what is the agent that is infecting you. Figure out what antibiotic works the best on it, and then give you that. That's the best situation. But right now we're not doing that. One more thing I was going to say that I forgot. Okay, so that's an issue. Education is obviously important. It goes along with it, and all of these have helped us actually reduce the bacterial infection. But unfortunately, it's coming back, as I told you, and a lot of people are worried about this. Now, we do find new antibiotics. There are a bunch of biologists who are in search of antibiotics, okay? And yeah.
No, I just to talk about that too. I guess a lot of drug companies are getting away from antibiotic research. The government can't find enough people who want to do antibiotic research. There's not enough money in it. So we might be coming to a bottleneck with the antibiotic.
Yeah, we do have an issue. And the problem is they're becoming resistant to everything you have. So then you need a different mechanism. Either you have to develop it in the lab, or you have to find it in nature. And so you have people who, biologists who are doing this, searching in the oceans. There are a bunch of animals that produce antibiotics, from frogs to ants to, I forget, lizards, all kinds of things that they found. And that gives us an idea for a new antibiotics that may be functional, that works. But if you're infected today and we don't have anything, then you're dead. Or, so that's the problem. It takes time. What?
Humans produce antibiotics?
We can synthesize by knowing its structure.
I mean, like bacteria.
Can we produce naturally antibiotics? Not that I know. I don't know any antibiotics. We produce antimicrobial enzymes, but not antibiotics. Like lysozyme is an enzyme that breaks down the cell wall and destroys bacteria, but it's not an antibiotic.
Is there any way that we could possibly, like, the pathogens that we try to kill, that become more resistant to antibiotics, is there any way that we, like the ones that we can become resistant to the ones that are resistant? So that we would actually produce different enzyme or different...
We don't evolve fast enough.
Well, we produce antibiotic antibodies, okay, when we get infected, and you have memory cells, and that's one way to do it. But again, these bacterial cells also change. And the first time that you get infected, if your body is not in optimum situation and is not protecting you against that, and you get sick, that means that bacteria is overcoming the situation. You do have a natural way of fighting bacterial viral infection. That's memory cell. We're going to talk about next semester. All of your white blood cells, your antibodies that you produce, and so on. But it's not always what? Successful.
Well, like the experiment they were talking about in this chapter 2, where they put bacteria on different mediums that didn't necessarily match their normal environment. They were able to very quickly adapt to it and evolve to it, so they can evolve way faster than we can even control it.
Yeah.
So it's always going to be a problem.
They would, in some cases, they could be ahead of you, the pathogens, and so that would be ahead of the system. But in some cases, we become immune to them because your immune system functions well, and then it doesn't cause as much problem. So
yeah, they're talking about trying to fight antibiotic-resistant bacteria with bacteriophages and viruses, so that they can move just as fast.
yeah, there's a good article. Some of you may want to present that next semester in Scientific American that talks about that. Yeah, there's all kinds of different now ways that people are looking. How do we want to attack? One way is to find new antibiotics. Other ways to maybe find other means to actually overcome infection. But we have got used to this idea that infection is not a big deal because you go and take some pills and now it's coming back. That's a big lesson. Okay. The other thing I was going to say that I remember, some other countries like Mexico up to last year, you could go buy antibiotics in, any drugstore, any store. So it was available to anybody who wanted to take it. It's changing, so those things are going to help. It has to be prescribed, it has to be monitored, and so on. All right, let's go quickly. I have a bunch of stuff to tell you in the lab also. Let's see. Where we are. Okay. Quickly, we already learned about this. 3 different ways that bacteria can become resistant to that mechanism that I want to give you, that we know they become resistant to a particular antibiotics. They definitely have the right gene in their plasmid that produces, maybe one way would be an enzyme that degrades and breaks down the antibiotics as soon as it goes in. It breaks it apart, so it becomes non-functional. Okay, that's one mechanism. Are you guys writing these down? It's not in your book. Number 2, or maybe it's in the handouts, I don't know. Number 2, they may have an enzyme that modifies the antibiotics. In this red piece, antibiotic goes in, they add chemicals to it, modification happens, this is the enzyme. That molecule doesn't work anymore. Okay? Here, it could be that it's pumped out. So these are different ways that they found bacteria can resist. Antibiotic goes in, instead of doing what it needs to do, it kicks what? It's kicked out, and it doesn't function. Okay? Also, we learned that, again, 3 different ways that they will pass these genes to each other. It could be that they pick it up, right, from the environment, through viruses, phage, or through conjugation.
Do you know if there's any way to kind of de-evolve a bacteria from its resistance?
Do what?
To de-evolve a bacteria from its resistance, so it's gained some kind of antibiotic resistance. Is there any way to reverse that?
Yeah, you can do, I don't know a specific experiment, but you can reverse the adaptant organism to something, especially bacteria. But then you have the other ones all over. You have to find a way to make these to take over that other group. But you can move through selection, you can move it this way or the other way. With things that propagate that fast. Okay? Let me give you one more thing and then we'll take a break. This is a quick, was this in your handout?
Yeah.
Okay. So let's give you 2 or 3 of these to remember, not all of them, just so that you get an idea about mechanism of action. Each one of these things that we talked about, it's a whole chapter in microbiology book, like this one is the whole chapter, right? Which is we've taken it. Different mechanisms that antibiotics work, right? Let's see which ones are kind of common. Things that you know that's kind of interesting biology here. All right, let's look at erythromycin. This is a common one, okay? Highlight that one. You have the handout.
Which handout is this?
Let's put it in your notes. And if you go to the outline and notes.
Oh, it is?
Was it there?
Yeah.
Oh,
Page
all
7.
right.
All right, very good. Erythromycin, okay? This is a common one that we use. What it does is stops protein synthesis in bacteria. Is that the type of question I may ask you? What does it inhibit? It affects ribosome. Remember, ribosome is made up of a large piece and a small piece, so it affects the large piece in this case. How come it doesn't affect our own cells? Because our ribosomes are different. Okay, that's the key thing. All right. So as you see, it's a bunch of those that have the same mechanism. They stop and inhibit the 5S, the larger piece. Uh, Tetracycline is another old one that we've used for a long time. I like that one, tetracycline. Again, protein synthesis is inhibited, but in this case it affects what? The smaller, the 30S. That's the S and number is sedimentation rate when they do centrifugation and they go down. That's how they refer to it. Okay, so you got already two. Tetracycline. Streptomycin is another one, same thing as the common ones used. And then a bunch of them here, where is it? In this group, penicillin, all of you know that one. Prophylactin is a group, penicillin. What do? They block the formation of cell wall. Okay? Now, the other option is doing synthetic drugs. You create synthetic drugs that some way attach the bacteria in a particular area. But it's not as easy as people think, making synthetic drugs. And sometimes it takes us years and years and years of research by organic chemists, people, pharmacologists, and so on who do research, try to design a drug. A lot of times nature has already created these things, and it's easier to get it from nature. Okay? But remember those. That's about it. Just to show you how a molecule may kill something. All right. I think I should take one more thing. Okay, short video and then we'll take a break, okay?
Along with all the problems that war brings, we're now facing a new enemy emerging from Iraq. Each of its soldiers are packing weapons, dozens of them. These guys can survive for weeks at a time without food or water. We don't know how to fight, but we've got to find out. Guns and tanks won't help us here. But as correspondent John Torres reports, what we really need is a good biologist.
There's a killer on the battle-torn streets of Iraq, but it doesn't carry a gun. It's attacking injured soldiers. With better armor and advanced medical care, they're surviving in larger numbers than ever before.
I was a doctor in Iraq with the Air National Guard, and I can tell you from firsthand experience, the survival rate for wounded soldiers, it's a remarkable story. But it's one with a downside.
That downside comes in the form of a tiny microbe with a powerful punch. Here's the culprit. It's a bacterium called Baumannii, referred to in Iraq as Iraqibacter. It's named for microbiologist Paul Baumann, who researched it back in 1968. But even he couldn't predict what this tiny single-celled organism would one day become. Like most bacteria, it lives in colonies and is constantly reproducing, simply by dividing and dividing again. A single bacterium can give rise to 5 billion trillion in only a day. This bug used to be relatively harmless, yet somehow it's found a way to transform itself into a drug-resistant killer. One of its many victims was ABC News correspondent Bob Woodruff. On January 29, 2006, while embedded with the U.S. 4th Infantry Division in Baghdad, his vehicle was hit by a roadside bomb.
We have some breaking news to report. Our co-anchor of World News Tonight, Bob Woodruff.
To keep him alive, doctors had to remove part of his skull and induce a medical coma. Miraculously, Bob was stabilized and evacuated to Bethesda Naval Hospital in Maryland. His wife Lee was there by his side. What was going on with Bob at that point?
He underwent many different surgeries for different things, but I think that's the point at which they became nervous about pneumonia and sepsis setting in. And in fact, that was what had happened.
A Baumannii infection had spread throughout his body, and he was back at death's door.
It seemed impossible to me that someone could be in a war and be hit by a bomb and survive this, and then be actually felled by a simple bacteria in the hospital.
Bob Woodruff is just one of many soldiers and civilians picking up this deadly microbe in hospitals along the evacuation chain out of Iraq and bringing it back home to America, where it's infecting even people who have never seen a battlefield.
It has this ability to hang around in places where it ought not, like on doorknobs and pillowcases and the like.
Where it can survive for weeks. So why not simply use an antibiotic like penicillin to fight it? After all, haven't antibiotics been the magic bullet saving soldiers' lives since World War II?
We saved a lot of people's lives. Penicillin was a wonder drug.
But something has changed. Now the bugs are fighting back. Microbiologist Mike Smith demonstrates how drug-resistant this bug has become.
You take Baumannii and you put it on a plate containing imipenem.
He places colonies containing millions of bacteria in several petri dishes and confronts them with imipenem, an antibiotic so strong it's nicknamed gorillacillin. After 12 hours, all the bacteria should be dead, but they're not.
Unfortunately, this is the kind of thing that we're seeing where certain colonies are surviving, and in this case you can see a few in the middle there.
Now looking at it, there's only 6 or 8 little colonies.
These are the only ones that are living, so the entire population now of remaining bacteria are imipenem resistant. So this is the strain.
Smith and graduate student Tara Janulis prepare a sample of Baumannii. Its DNA will be sequenced, one letter at a time. The results reveal that Baumannii has large sections of genes that don't belong. Foreign genes that are giving it resistance to antibiotics.
There is a multi-drug resistant strain that took 45 different drug resistance genes and stuck them in one spot. This should be alarming because that's what this bug can do.
How is this possible? There's only one way we get our genes, and that's from our parents. It's called vertical gene transfer. But it turns out bacteria can also get genes in a process called horizontal gene transfer. One way that happens is when two bacteria get together for a little friendly conjugation, the microbial form of snuggling. They form a connection and squirt DNA into each other. Turns out Baumannii has been getting a little too friendly. Could that be what's making it so nasty? To find out, Mike Smith zaps a colony of Baumannii with electricity, creating over 1,000 mutant bugs, each one different, each one missing one known gene. He takes these mutant microbes and feeds them to some microscopic worms.
They're an amazing little organism.
For Mike, one of the best things about these worms is that they love eating bacteria.
That's what it eats for breakfast, lunch, and dinner. And it turns out that when these worms eat disease-causing bacteria, it often kills them too.
Just like lethal bacteria can kill us. But if Mike has succeeded in zapping out the lethal genes, then some of his worms should survive. Will it work? He'll find out at dinner time. The worms that survive give Mike the information that he needs to know who Balmanii has been getting friendly with. It turns out one of the deadly genes comes from a well-known killer, the bacteria responsible for Legionnaire's disease. A deadly bug first recognized back in 1976, when, at an American Legion convention, over 1,000 people suddenly came down with a serious lung infection. 34 died.
It's taking in drug resistance gene and it's developing drug resistance. It's taking in virulence or disease-causing type genes and it's using them.
With the addition of some new genes, Balmanii becomes more lethal and harder to defeat all at the same time. It's an ongoing battle, not easily won.
Perhaps instead of trying to just overwhelm them and kill them, maybe we can coerce them into doing something that they weren't thinking about doing if we understand how they communicate.
But bacteria have been around a lot longer than we have, and they are very good at adapting to anything that we throw at them. So for now, it's a matter of early intervention, better hospital hygiene, and good medical care. That's what saved Bob Woodruff.
He's had, I think, a miraculous recovery. It's pretty phenomenal. It speaks to the acute medical care that he got in the beginning as well.
All right, did you guys notice the attitude that she had towards the race